two books in the entomological collection of the Zoology Research Group, University of Silesia in Kutsk, Poland. Agnieszka.
Yes, good morning. My name is Agnieszka, Bugaj-Nawrocka, and today I'm very glad to be there, and me and my colleagues, probably you know them, Artur, Taszakowski, Professor Gorcyzca, Professor Chłond, and Professor Wierczorek, we want to use this opportunity to present you our Trubax collection, which is now available for a wider group of scientists, because we make some order with it. But first of all, let me start a brief history of our institute and how this collection just was created. So University of Silesia in Katowice was established in 1968, and as our region is really industrial, so there was a big need of biological studies, especially related to problems of environmental protection. And the Institute of Biology was established in 1969, and the organizer and the first director of our institute was Przemysław Trojan. Initially, there were only 3 departments in our institute: ecology, biochemistry, and microbiology. And in 1973, the Faculty of Biology and Environmental Protection was established on the basis of the Institute of Biology, and then, among others, Institute of Zoology was established, and 3 years ago, in 2090, after big modification, now we are zoology research team within Faculty of Natural Sciences in Institute of Biology, Biotechnology and Environmental Protection. But no matter the names of the faculties and the institutes, our team, our researchers works with Hemiptera groups more than 50 years right now. And we collect the material almost all over the world, but as we live in Poland, mostly from the Central Europe, but I will talk about this a little bit later. So our scientists conducted zoonotic, faunistic, zoogeographical and taxonomic research based on selected group of insects in the order Hemiptera. Now we also have 2 phytotrons in which experiments with aphids mostly are carried out. We also have amber laboratory and the breeding of exotic animals where we have mostly insects. Years of research on insects allow us to collect some unique resources in the form of collection, which now counts more than 100,000 specimens of Hemiptera, and they are divided into 5 collections. So we have a collection of aphids, scale insects, psyllids, Auchenorrhyncha F and Heteroptera. All those specimens can be found in showcases, boxes as microscope slides, or as inclusions in amber. In 2018, we started a digitalization program which was called Hemipteron. And thanks to this program, we have the opportunity to make some order and make some cleaning with our collection. So in this collection, special place is occupied by collections of specimens which were prepared by our Polish eminent specialists like Professor Koteja, Professor Klimaszewski, and Professor Szeregiewicz. If some of you work with collection and know how to digitize the material, we do this the standard way. So we accurate identification, we segregate the material, then specimen data were entered into the database. Each specimen have ID number, so we can easily easily find it in database, and all slides were scanned, and the specimens on pin were photographs as well as those in amber. This is just how look like our database. So we try to possess as much information as we could from the labels. So the standard information like who collected, where, when, who identified it. We also have 2 columns with actual names and the name which is on label, because as you all know, those names sometimes change, as we are all taxonomists, so we know how it works. So we try to always have the actual name and the name which was on the label when it was identified. Also for such groups as scale insects and aphids, we have information about plants which associate to these insects. In the case of Heteroptera, if such information were available, they are also in database. Just a few words about collection, because there is a lot of information. There is no time to present it all, but this is rather small collection, we know it, but we hope this will be a good supplement for other big collection in Central Europe. So right now we have 34,000, more than 54,000 specimens which are already digitized, more than 6,000 wait for their time, and collected specimens represent 40 Trubax families. Here you have just easy graph to show you the numbers. So of course mirids are the most numerous in our collection. Then we have pentatomids, Nabide, Ropalide, etc. As you can see, some of those families are rather poorly represented, but this is due to this, to the geography, to the place of Poland in Europe. So those groups sometimes are just not present in Central Europe, and if they are, we just have a few specimens, but we are try to make it most numerous by to possess some specimens. And here you have a map. You can see not the regions, but the countries from which we have the material. So, as I said, most of the material is from Poland, from Czech Republic, Germany, Slovakia, so there's Central and Western Europe. As we, as a researcher of zoology team and our students, which prepares their master thesis and their PhD thesis, they collect material in field, mostly in different parts of Poland, but also in some parts of Europe. But also, we also have material from Asia, Africa, and South America, and they were collected by our researchers during their research expedition. Here a simple graph just to show you the most numerous group in our collection, how many genera and how many species they represent. So, as I told, mirids are the most numerous, then Pentatomidae, and a few other groups. Of course, we have type material, not a lot, but still there is about 195 specimens which are type material. 105 are redovids, and 88 are mirids. Those from mirids are mostly type material of Professor Gorcyzca from Cylapines and the other group of mirids. And thanks to the program Hemipteron, we have the opportunity to create a website. And so we put there information about the main group of Hemiptera for people who just want to know something more about them and they are not maybe a specialist. But also the most important is that we have there our database. So you can search information about the specimens which are in our collection. You can use filters, so you can just search for us by the name of the species, or maybe select some other filters like year, place of collection, who collected, etc. Yeah, and we hope that, as I said, this is not a big collection, but we hope some of you maybe find some interesting specimens for your groups to study it. There is also still a lot of material which is undetermined, or even if it is determined to rather to the genus level, not the species level. So we will be very pleased, and it will be very good for our collection if some of you could just borrow the material and identify it, so our collection will be better in that way that we have identified species in it. Yeah, so that will be all about the collection. Thank you very much for your attention. I just want to say something. We prepare publications about the specimens in the collection, so much more information will be in those publications. We want to make 3 of them. First, probably, I hope, will be finished at the end of September, and this will be about Pentatomomorpha, all Pentatomomorpha in our collection. The second one with Geromorpha, Nepomorpha, and Cymicomorpha, but without mirids, and the third only with Miridae family. So I hope you will find more information, more details in those publications.
Questions?
Collection manager here. So are you going to put all that data on, I think UNIS has a data set, which is the European biodiversity website, that they put their data online so it's available across the world. I know GBIF does that too. Are you going to be putting the data on that? Because in previous institutions where I've worked, when I put the database data on GBIF, our loan requests went up like 3 times. So,
yeah, so our database is just prepared as they want it in GBIF and other databases. So we have the same
the Darwin core, yeah.
Yes, the order of the database. So we want to just put it to the GBIF and other databases, so it will be more visible for others. Yes.
It's a cool collection. Good job.
Thank you very much.
Dear colleagues, there's a slightly change on the program. Lanna Cheng is going to present her presentation before, and she's also going to give a short review and biography on Niels Moller Andersen at the same time, because we are going to start the symposium on the Geromorpha in memory of him. Thank you very much. Lanna, please.
So now I have a little more time to say something about Niels Moller Andersen. For those of you who are young heteropterists, you probably never heard of Niels's name. But for all of us old members, we have been seeing Niels for many times, many IHS meetings. So this is the a picture of Niels published in his official obituary from Denmark, and the obituary was in Danish. Niels was a Danish scientist. He spent all his academic life at the University of Copenhagen looking after the zoological collection. In those days, it's called the ZMUC, Zoological Collection at the University of Copenhagen. And he tried to build up the collection in the museum, especially on Heteroptera. His own interest is on, I press the lower, which one do I press for the next slide? This one, yes? I do Mac, so I'm not very good at it. So his love is actually on Geromorpha rather than Nipomorpha, and one of his most important contribution is this book. As you can see, it was published in 1982, and it has almost everything you ever wanted to know about this group. It has detailed studies on everything, and you can see a summary. And then at the bottom right, left, can you see that little whimsical cartoon? So he realized after publication of the book that there are still a lot of things remaining to be learned, okay? So apart from this book, there's another book that should be there. Anyway, he has done all the illustrations by hand in the book. As you can see, all the details are presented here. There were no fancy attachments that would allow you to trace the color pattern or details. These were all his own original drawing. He was a really fine scientist. Apart from being a fine scientist, he's also a very good artist. Okay. Apart from that book, he also published a book on the Australian water bugs with Tom Weir, and the book was published after he passed away. So on the right-hand side, you can see the summary of his contributions to the field of Geromorpha. Apart from all the papers he has published, which are more than 150, he has described more than 200 new species. And then there are the taxa named in his honor. You can see all this. And this can all be found in his obituary published by Spence and Damgard, and that was published in 2008. So as you can see, his contribution to Geromorpha is really phenomenal, and he has built up a wonderful collection of Geromorpha for the museum collection. I was introduced to Niels in 1969 by somebody who was a mutual friend. After I got to know Niels, he fell in love with Hellobates because it would give him an excuse to go to all the very interesting islands for him to collect marine insects. Apart from Hellobates, which is a marine gerridae, there are also other families of marine insects. So He traveled all over the world to collect things. But the two of us never wrote any paper together. We corresponded and we became very close friends. And then the last time I saw Niels was in St. Petersburg in 2002. He brought his research student, Jacek, with him. And at that meeting, he said, Lanna, we have never published any paper together. We talked about it, we have co-authored paper. Let's do something. I said, okay, fine. The Gerardi Halobates needs to be, the review needs to be upgraded. The most important thing was the classical key to Gerardi, Halobates was published by Herring in 1961. It's completely out of date. So he agreed to do that. And then he said, okay, I'm going to do the key. I said, all right, sign. It will be Anderson and Cheng. He said, no, you have to be Cheng and Anderson. I said, no excuse. Alphabetical order. Your name starts with A, my name starts with C. So that's it. So you can see then the news obituary, if you want to find anything about his publication, it will be in the book, in the publication on the top, and then I wrote something about Niels and Halobates at the bottom. So now I will start my own presentation and tell you about what, something about the distribution of Halobates. So most of them may not have heard of Halobates. It's a genus in the family Gerardi, and it is only the only species that has a genus with species found in the open ocean. So we all know, without any doubt, that insects are the most numerous, abundant animals in the world. A conservative estimate is we have about 5 million species of insects in the world. And then among that, there's a little bit that are found in the marine environment. Actually, among insect orders, more than half of the orders have representatives in the marine environment. But when you go to the Heteroptera, Hemiptera, you have a little tiny sliver that represents Halobates. There are just under 50 species of Halobates known in the genus, and it happens to be the only genus of insects with representative in the open ocean. Not only the only insect, but only animal living in the open ocean far away from land. No other animals can live in that environment. There are surface living like a Physalia, Portuguese man of war that gives you a very nasty sting, but it doesn't actually live at the surface of the ocean. Halobates does not dive and do not swim, and it was discovered in 1822, exactly 200 years ago. Don't worry about the description. Very few Germans can even read it because it's written in cursive German. So this person was given a series of samples collected on an oceanographic cruise, a Russian cruise around the world. He described something like 82 species of new insects, and from land as well as from the sea. So Halobates remained something which is odd. People collect them, they were described. And then this is what it looks like. On the right-hand side is a live animal. As you can see, it's completely wingless. It has never got wings at any time of his life. And the adults measure about 5 to 6 millimeters. So imagine you go to sea on a cruise ship, you see whales, porpoises, you see fish and seabirds. Can you spot any Halobates? Never. It's so tiny, it's the size of a black peppercorn. So even Darwin on his cruise on the Beagle around the world never saw it, although it was described 5 or something years before his cruise. So here is a summary of what Halobates is. And then most of I told you about just under 50 species. Most of them occur in the coast associated with mangroves. And this one shows you the map of all the key locations of the type species found. So you see where the coastal species are located more or less in the Indo-Pacific. There is nothing, not a single coastal species found in the Atlantic Ocean, and the closest one we found is in the Galapagos. There's an endemic species and also one in Hawaii, and the rest are found in the Indo-Pacific. What about the open ocean species? They are found, I told you, in the open sea. How do you collect them? You have to go out on a research vessel. And this one actually is on the Beagle cruise. In 2001, they tried to repeat Darwin's cruise, and we did collect some halobates. And they are found on the surface. Most biologists who go to sea to collect specimens, they use a net that goes down to the ocean. So we have to get a special net that skims over the ocean surface to collect them. Because if the net is going below the surface, you don't get them. When you get a plankton net, it comes up, you get 1 or 2 specimens. So this net was specifically constructed to collect halobates. Okay, this shows the distribution of the 5 open ocean halobates species. So it has the widest distribution of any insect in the world, I think. So look at that. In the Pacific Ocean, all 5 species are found, different colors. In the Atlantic Ocean, only 1 species. And then in the Indian Ocean, 2 species. It's a very interesting distribution pattern. So what I'm going to tell you is 3 studies done to figure out their distribution. And as you can see on these 2 phylogenetic trees, the one on the left was the first, based on the first genetic DNA sequencing study on halobates. And on top, of the one that highlighted, micans, splendens, sobrinus, and then below, Sericius germanus. These are the 5 open ocean species. They are more or less sort of related. They may be 2 separate origins of the open ocean species. But on the right-hand side, on the paper published 2018, they were not highlighted, but you could see that perhaps instead of 2 origins for open ocean habitat, there may be 3. So we still do not know whether the open ocean ones came from 1 origin and then they speciated, or 2 or 3 or 4. So this has to be investigated later. Okay, now we get to the distribution of the cosmopolitan species Hyanus. This is where we find them. On the broad scale, we say that it's found between latitudes 40 north and 40 south. But in the Pacific Ocean, it's only found in the middle. In the Atlantic Ocean, there are no other species, so it's found all the way from 40 north to 40 south. In the present day, there is no way they can go around the Horn of Africa or South America, because the sea temperature has to be above 20 centigrade for them to survive. So, are the 3 species populations in 3 oceans different or not? Morphologically, you cannot tell whatsoever. So this was done in 1980. We got some specimens, a few specimens. We did the CO1 marker gene sequences, and as you can see here, the 3 are not really specifically different. They are still quite similar. The Pacific Ocean population is more similar to the Atlantic than the Indian Ocean, which is quite different from what I suspected, because they could have moved from the Indian Ocean to the Pacific, but they are prevented to move into the Atlantic Ocean because of the closure of the Isthmus of Panama, right? Even when they opened the canal, they couldn't go across. So this is a little interesting distribution. The next distribution we look at is the species that's found in the North and the South Pacific. So how did it become separated in the middle? Unfortunately, is no data on surface ocean currents for me to correlate to their distribution, because oceanographers, when they study currents in the ocean, they measure currents from either 10 meters down or 1 meter down, but nobody could could tell me what happens at the ocean surface, the skin of the ocean, where Halobate studies inhabits. Now we have better system to measure that, but there's no analysis yet. So all we could do is to, find out whether or not genetically they are different. So again, we got some specimens from the north and the south, and we used 3 genetic markers. By the 1980s, we progressed from just 101 to 3. So I'm not going to go into details of that, except with those 3 genetic markers. And the interesting thing is that there are the two populations are still clearly the same species. There's very little genetic exchange between the two. So they must have been separated for although they have been separated for tens of thousands of years. So what we then, so that is what it stands now. And because the two populations are still separated, it means that the ocean currents have remained stable for at least tens of thousands of years for the two populations not to mix together in the middle. Then the next one we want to figure out is look at the right-hand side of the Pacific Ocean. That's the one in the middle. You see the very dark splotch. That is the area what we call the Eastern Tropical Pacific Ocean, where 3 species, where theoretically 4 species of halobates can occur. The 5th species that is found in the Pacific, the 5th open ocean species is Halobates gammanus. It's found in the little black dots in the Red Sea all the way to the Pacific Ocean, and it doesn't go any way beyond Tahiti. But in the Eastern Tropical Pacific Ocean, we theoretically could find 4 species, but in actual fact only 3. So these are Mycans, Sobrinus, and Splendens. So I wanted to find out whether or not, how is it that they are, the two species are only restricted to the Eastern Tropical Pacific. Theoretically, there are no physical barriers in the open sea. They could walk or skate, they skate. They could skate from the coast of Americas to Australia, Japan, Singapore. I come from Singapore, so we wanted to do something about that. So I persuaded a friend who was on an oceanographic cruise that sampled this area from the coast of Mexico on the right all the way to past the Hawaii and then back. So look at all those black spots. Each spot represents one station they collected halobates. So this was really a very nice collection, and we were able to get the 378 specimens. You can see on top how many hundred specimens we have collected. So it's an unprecedented number of specimens collected by a deep net singly, and they were all preserved in ethanol. So they are a very valuable, clean collection for us to do molecular analysis. So now we have come to the 2000. We are able to do molecular analysis cheaply and quickly. When we started in 1980, each specimen cost us $10 to begin with. And then by the time we did this, it's probably about $1, I think, per sample. So this is the summary of what we have found. Each of the species had a population which remained as one population. There are no subspecies, no evidence of any subspecies or so. And then the genetic diversities implies some different evolutionary trajectory. And then I will show you some map of the skyline plot to show that, you see this, there are many haplotypes, but there are no subpopulations. So then why are these two species, the black ones, why couldn't they go away? So at that stage, I tried to contact a paleo-oceanographer who specializes in ocean circulation systems. Because the mycans, which is going all across, that's okay, no problem for us. But these other two species, why couldn't they spread out? So when he looked at the map, he said, oh, fine, no problem. This is what happened. So you can see then here, on the right-hand side, the sobrinus and mycans, they showed population explosion, respectively at 50 and 10 on the bottom one. Oh, it didn't show, right? It's like 50 and 80,000, 100,000 years ago. But for the sobrinus, it's about 1 million years ago. So there is a period where the population exploded that long ago. So the period of population explosion of the two species, mycans and sobrinus and splendens, are quite different. And the sobrinus and mycans happen to have population expansion during the period when the ocean system went through an El Nino period. So the seawater temperature went up, it was warm. And they are then more adapted to the warm seawater temperature. But it's very odd that for splendens, which had a population explosion 1 million years ago, and it corresponded to a cold tongue, which is you hear about the Peru current, which is cold current, and then because the deep ocean water will come up with upwelling, and that brings a lot of nutrient. So there's a lot of food on the surface. That's where you get your tuna and your sardines. So this is the connection. So this is the evolutionary history according to paleoceanography. So now, why do they not expand their population beyond this area? We collected the samples from the coast to like about 7,000 kilometers away. But these two species, sobrinus and splendens, never went beyond that area. Why do they not go beyond? We are not sure, because we do not have any oceanographic data to support this. So at the moment, this is where the story stays. There's a lot of work still going on. Now some active research are carried out at the King Abdullah University of Science and Technology in Jeddah in Saudi Arabia, and the other active group of researchers, research projects done Halabatis is at the National University of Singapore. So the next speaker will tell you a little bit about what is being carried out in Singapore. And this is the end of my talk, and I will try to answer some questions. What they doesn't want to give me the last slide. Oh. It's okay.
Thank you very much.
The last slide is just a picture. So if you have any questions, I will try to answer.
Looking at the map of the world distribution of Alobatis, one noticed that they are present south of 40 degrees of latitude in all oceans.
To look at the map?
No, well, it is not necessary. So they are present in the Atlantic Ocean, just at the entrance of the Mediterranean Sea, but apparently they don't penetrate it. Do you think that they could be present, also considering that there is a fossil species found in Italy, Alobatis rufoi, or they could enter the Mediterranean with the warming up of sea temperature now? That's
Very good question. Why is it found in the Mediterranean? Yes? Okay. You are right. The fossil of Alobatis was discovered on the coast of Verona, now in Italy. So that was a 45 million year fossil. 45 million year old fossil. And we could not do any DNA on that. I would dearly love to find out whether Alobatis now can be found in the Mediterranean. The Mediterranean Sea was completely drained. And then when it refilled about 6 million years ago, it's quite a fast refilling. After it was refilled, the water was cold. So Alobatis could have migrated through the Straits of Gibraltar to the Mediterranean, but if the water is less than 20 degrees centigrade, they could not survive. So the Mediterranean Sea, actually in the summertime, it may go up to about 18 degrees. Now with climate change, it is getting warmer. And there are little pockets of areas where they may be able to survive. So I have encouraged my colleagues in Europe to try to make an expedition in the Mediterranean to see whether any in some areas they could appear. They could really come through. And another possibility is that eggs are laid on solid surfaces. So they could be attached to hull of ships, right? And then as the ships go through Suez Canal from the Red Sea and all that, they could bring Alobatis back. So please look.
Well, that was amazing. Thank you very much. And I have been fortunate to see some of these in New Guinea once, and I was so excited when I saw them. But I just have some general questions, because I don't really know much about how they manage to actually survive in these conditions. So can you give us some insight in how first they might deal with sort of uneven surface in the ocean? So when it gets stormy, for instance...
You have too much echo.
Oh, sorry.
Okay, I can't. Okay.
So can you give us some insight into how they manage to survive in sort of turbulent waters in the ocean, like when it gets stormy? And also, how do they survive in sort of high salt conditions? Do they have special salt tolerance abilities?
I'm not quite sure about your question.
So the two questions is how do they manage to survive in turbulent waters? So when it gets stormy in an ocean, for instance. And the other one is about salt tolerance.
Salt tolerance.
Yeah, so how do they manage to survive in high salt conditions compared to, say, other bugs?
I don't think insects have problems with salt secretion because of the Malpighian tubules. They do drink water and then they can just get rid of it, right? Mm-hmm. But the biology between the open ocean and coastal species are actually quite different. Mm-hmm. They do drink water, and the coastal species, open ocean species will not survive if it is, you give them salt, fresh water for 2 days. Mm-hmm. Yeah. So, but in the open ocean, they do not have problems because even if it rains, you get mixing very quickly. So there's no problem with that. And you have a second question.
Yeah, the other one was about turbulent waters. So like in an ocean, for instance, you get big storms. Yeah, so how do they manage to survive in something like that, where you've got like huge swells?
That's the first problem in the storm.
Yeah.
What happens? Think of little peppercorns in the water, and on the open ocean you get big storms coming. Okay, so I didn't go into the biology because they have they have very tiny microtrachea on the surface. So they have, all insects are air breathers. They could not really go underwater for more than a certain number of minutes and they would die. So in Helobates, they have tiny little microtrachea, about 1 micron high, and these trachea are lined up like this, like little mushrooms, and then under this layer of air is trapped. It's called a plastron arrangement. Theoretically, when they breathe underwater, oxygen level goes down. It goes by osmosis from the seawater. It goes in, so they could survive this way with no problem. And what's more, they are also super hydrophobic. So the seawater, the rainwater, as it hits them, rolls off. So there's an article in PLOS Biology. If you Google it, we explain some of these there. So but the work is still going on. How long they could survive underwater? I know, submerged for up to 18 hours. Then I was too sleepy to go on with the experiment. But they do not drown. They just pop up. They're like being wrapped in a air bubble. Yeah. So they just come up, back up, unless if you use boiled seawater with no oxygen, they sink down.
Wow.
Okay.
Amazing. Thank you.
Thank you.
Do the open ocean species and the coastal species have similar behavior in this way? You know, when they're like, can the coastal species also survive underwater like that?
The open ocean and coastal species are very different in biology in many ways. They are physiologically, they're morphologically alike. The freshwater, the coastal species would lay eggs on solid material, tree trunks, rocks or something. Open ocean, nothing. So they have to be able to find something that's floating and lay eggs on it. So with the plastic pollution, floating bits around, so there was an article to say Helobates are going to take over the world. Another thing is that because they live in the ocean, they have to have membranes to deal with maybe salt solution and all that. So they have in their structural lipid some long-chain fatty acid that may have something to do with the formation of membrane that deals with salt water. The fresh coastal species don't have that. And furthermore, living in the ocean, you have to apply suntan, wear a hat, okay? What are they going to do? No shelter. Coastal ones are associated with mangrove. Midday sun, they're under the mangrove. So what do they do with it? We are doing some work on that at Saudi Arabia. We don't know yet. Stay tuned.
Very cool, thanks.
Thank you very much. Gracias.
Life story, storing and reference sequencing, and marine geomorph. And Genji.
Right, thank you. So I hope you can hear me. So thanks Lanna again for the great introduction on Helobates. Makes my life a lot easier. It's just me and my presentation before we go for lunch. So I'll try to make this as painless as possible. Okay, so I work on marine insects like Helobates. So let's get started. So a lot of you here will probably be taxonomists. I, for one, am not. Full disclosure, I am brand new to the world of insects. So I learned that species descriptions for insects are actually based on male specimens, in particular the male reproductive organs. So you can see the panel on the right over there. The rows are species, and the columns are the different life history stages. So it's quite clear that when you put them side by side, that nymphs and females don't really look like males. And this was something that I didn't know, and I initially thought I found a new species. So you can imagine how disappointed I was. So actually, without accompanying males, without anatomy of the male reproductive organ, it is actually quite difficult to perform species identification for aquatic insects. And I would imagine maybe other insect groups as well. And so you know that Halobates, they actually live on the ocean surface. And so behaviorally, the different life stages actually exhibit so-called swarming behavior on the 2D space. So the nymphs tend to group together, and the adults tend to group somewhere else further out at sea. So this makes life, for me, a new researcher, a lot more difficult, I would say, because like chances are when I go to the coast, I collect insects, most likely there'll be nymphs, and then you know, I just can't identify them. So something that I'm working on, which is DNA barcoding. So I'm more familiar with lab skills. So actually, in recent years, a lot of people have been doing DNA barcoding for insects, and you know, DNA barcodes are just these short fragments of sequences that can help identify your specimens to species, hopefully. So anytime we go out to collect insects, it's something that you'll see on the right. It's a falcon tube just full of ethanol, and there's this mass of black stuff, you know. So when I first started, you know, it was quite tedious. In fact, I think I took, later on I'll show you the actual sample size, but I took like 2 weeks to sort them out alone. So what I am actually applying here is actually DNA barcoding, which is I'm using it to sort my samples to species. And this is in scientific vernacular, it's called the reverse workflow paradigm by Wang et al., which is you sort your insects first with sequencing, then you find out how many species you have. Traditionally, one would normally sort them first, pre-sort them, and then they pick out select representatives for DNA barcoding. But now, again, like as Lanna has mentioned, it's quite cheap, so we can do it quite easily nowadays. So what I did was that I applied third generation sequencer. It's what you can see, it's called the MinION. It's actually a handheld sequencer. You can actually bring it out into the field to perform your lab work if necessary. So I imagine it will be quite useful for like ocean faring cruisers. And I hope that maybe I can do it someday, now that COVID's dying down. So these are like my project objectives. The first one was, of course, to sort the different life history stages with DNA barcodes. And then later on, I'll talk a bit about this thing that you see on the screen here of disjunct CO1 databases and how I sought to bridge them with mitogenome assembly. So for this work, we actually did a collection of 6 sites across Papua New Guinea. And in the table on the top left, you can see that over here, 615 of the specimens were actually nymphs. So that amounts to about 80% of our collection was just nymphs. And out of the 6 localities, there were actually 2 localities over here that did not have accompanying males. So traditionally, these 2 localities might be excluded from a biodiversity study because there was simply no way to identify them. So we actually performed DNA extractions for all 757 specimens. We ran a PCR, and then we combined everything, and then we sequenced across 2 runs on a MinION flow cell. So in sum, we actually retrieved 738 barcodes, amounts to about 90% sequencing success. And so then we put them through computer programs to help delimit species. The programs are called ASAP and PTP, and we actually recovered about 10 species units. But unfortunately, when I match my sequence data to whatever was available online, actually what I found was that I could only identify 1 species. now. So why is that? Because I know from past work that helobettis has been featured a lot in phylogenetic studies. So it's a bit technical, but just bear with me here. So the reason for this is that it's actually the result of using two different primer sets. So the one that I use, Amplify, compared to a full-length CO1 over here, the one that I amplified was at the 5' end, or the front half, and the one that normally researchers use for phylogenetic studies is actually at the back half. So you notice that actually there's a break here, meaning that whatever my sequences that I produce, when I match them online, they will not match anything in this region. So actually, to add on, beyond just life history sorting with DNA sequencing, what we sought to do was actually to assemble the full-length CO1 gene, so as to try to bridge these two databases. So to do that, we actually took representative specimens from each of the species clusters, and then we perform low coverage whole genome sequencing. And we generated at least 15 mitogenomes, and we managed to recover full complete circular mitogenomes from the different species units at about 22 to 260 times sequencing coverage. Then thereafter, I extracted 3 mitochondrial loci, as well as the 28S nuclear loci. I chose these 4 genes because they were previously used in geromorpha phylogeny before. And so I produced the tree on the right. It's a bit small, but most importantly, it's my samples are the ones that are in blue color, and these are used for actually comparison with past data sets for species identification. So this is just a summary, the same figure from the past. So the one in the black lines, the discrete lines, we managed to actually identify 4 helobettis species with morphology as well as DNA sequence data. The one with the dashed lines are actually only identified based on morphology alone, because there were just no sequences available for these species in the past. And this poor soul over here, we only had 3 specimens, all nymphs, so we were unable to identify based on morphology, and no sequences were found online, so we put it as Helobettis SP. So some general conclusions. What we found that was that actually now it is easier and faster to sort specimens with DNA barcodes. Someone who is completely new to insect science, like me, I managed to do it. So I think it's a good start, you know, and the taxonomic expertise can actually be directed elsewhere. So just to focus on, so we went from 757 specimens to just about 9 or 10 species units. So then the taxonomists can come in to look at actually just 9 to 10 species units. So I hope this saves you some time and work. And we are actually also now with DNA barcodes, we are actually able to incorporate information from nymphs as well as females. So back to this diagram again, actually the Helobettis peronis that we found was actually just a single female specimen in our collection, which we probably might have missed if we had done just morphological sorting. So, so we are able to actually retrieve full-length mitogenome sequences now, and I think it's still a work in progress. I'm still collecting more specimens, and we are slowly but surely improving at least reference data sets for Helobettis in general. So this is just a short update. The work that I've just showed you previously was actually done a couple of years ago, and so now it's just give you a slight update. You know, since then, nanopore sequencing technologies have continuously improved, and the way you analyze or generate your DNA barcodes have also improved, which is actually the screenshot on the right is actually a screen grab of the program. it's a drag and drop, so you literally just pick a files, drop them into the program, and it will do the DNA, it will call your DNA barcodes for you. So things are getting a lot more accurate nowadays, and things are also getting a lot faster. With this program, I was able to actually retrieve more barcodes from the same data set, same set of reads, in just about 5 minutes. So things are definitely getting more convenient, and hopefully these technologies will help improve your workflows when working with insects in future. So I just like to acknowledge these organizations for funding my research, as well as my travel for this conference, and that's it. I'm done. Happy to take any questions.
Questions?
You mentioned that they have swarming behavior. Do they use some pheromones? Could you tell a little more about that?
We're actually not sure how they recognize each other by swarming, but they do swarm. We recognize that they do swarm. Primarily, it's also to avoid predation. The adults, in times of a food crisis, have been known to predate on their own nymphs. So the swarming behavior by nymphs actually helps to keep them alive.
Mm. Okay, so they probably swarm by just seeing each other, not by smelling each other or something like that?
That one I'm not too sure.
Okay, good. Thank you.
Thank you very much.
Thank you.
So I guess most of you are hungry. So now we are going to go to the lunch place. We'll be following Marc and me, so we go together as a pack, which is easier not to get lost. It's around 10-15 minutes maximum by foot.
I wanted to thank to Letitia Neri, who opened the chairperson duties here. She's new in the affair, so thank you very much for that. You did a splendid job. Splendid job. Also, I want to thank all of you who have adjusted to the preview time. This is very valuable because it makes it confident that we arrive on time every conference, every event. So it gives not relaxation, a spirit of calm and everything, a time for everything and a thing for every time. So everything is just matching as preview. Thank you for that. And then those who have presentations for this evening, I have all of them, but we have now some spare minutes. If someone wants to give me the presentation, I can upload to my computer, okay? Thank you.
Also, just before we leave, so it's kind of easier to get you this way than go around. If anybody has any food intolerances, food preferences, vegan, vegetarian, let either me know directly or the waiters at the restaurant, because there's menus prepared for everybody, and we do not wish anybody to get the wrong meal. And bring the tabs. If you do not bring that, you will not get food. They have that in order, so there's not additional people coming in the way. Thank you very much, and see you now. You don't have one. I guess las bosses. Pregunten si las bosses les poden deixar. Podem deixar les coses? No quedarà tancat. The place is gonna be closed, so in case you want to leave your stuff, queda tancada la porta ya. Vale. Yeah. The room's gonna be closed, so the door is gonna be closed. In case anybody wants to leave their stuff in here, it's gonna be safe.